show kar देंगे. Today we are going to talk about this anti-oncogene called p53. So this is often referred to as guardian of genome. Guardian, it takes care of human genome, which we are talking with reference to human. It takes care of our genome. It is protective in action. It is called p53 because of its molecular weight, which is 53,000. So this 53,000 molecular weight anti-oncogene, it protects DNA, but protection from what? Protection from molecular oxidants. DNA is prone to a number of damages which are occurring, which number to about several thousands per day. So many assaults are there which are capable of damaging the DNA. Radiation, chemicals, free radicals, and not only these cause assault, even as innocuous a thing as sunlight, that can also change a base. So change in the base implies change in the genetic data bank information, and that is serious. And as I said, as innocuous as sunlight, the damage also may be as innocuous as seemingly innocuous as deamination of cytosine to form uracil, or there may be extensive DNA damage by some of these assaulters. Now, it is serious because DNA is repository of all the information in our body. So change in the information can result a number of diseases, but among them, the most prominent I'm going to talk about are cancers. So in a way, we can say that P53, it affords protection against cancer. And that is actually one observation that confirms this finding, that majority of spontaneous cancers arise due to some crippling mutation of p53. More than 90% of cancers of all varieties. So it has got anti-mutagenic action. So this small 53,000 comprising of 393 amino acids, half-life only 5 to 6 minutes, how it protects against the DNA damages we are going to talk about. So just, as we said, radiations, chemicals, free radicals, among others, and our DNA is like a sitting duck. So many, firing from all sides. So जब कभी भी damage हो गई, the first thing you can visualize that damage must be repaired. So repair is required. P53 does that. It drives expression of a number of genes which code for enzymes of DNA repair. And then apoptosis, we are going to talk in details. Apoptosis means programmed cell death. When DNA damage is so extensive that it is beyond any repair, it cannot be repaired, then the best remedy is that the cell commits suicide. It quits the scene. That is beneficial for the body because a cell with damaged DNA is a source of trouble. We are going to see that also. At third position, I have written cell cycle arrest, but it is very important, should be at the first position. Just in this rough diagram, red color ka DNA, iska matlab I have shown that there is damage to the DNA, damaged DNA. Now, DNA is not only repository of genetic information, it also replicates. Replicates के बाद mitosis होगा, one cell divided to two to four, like that. तो अगर डैमेज डीएनए हो गया, इफ इट गोज टू द प्रोजेनी, दैट इज वेरी सीरियस। तो रिपेयर होना जरूरी है, बट द सेल साइकिल it occurs after a limited cell division occurs after a limited period of time. I think 24 hours. 24 hours. You can see here complete cell cycling about 24 hours. तो ये 24 hours में रिपेयर से पहले ही अगर डिवीजन हो गया, the purpose will be defeated. So it is necessary that there is a halting at this stage. माइटोसिस होने से पहले ही रिपेयर हो जाए, तो उसके लिए या एपोप्टोसिस हो जाए, उसके लिए सेल साइकिल को अरेस्ट करना जरूरी है। आई एम टॉकिंग रिपीटेडली, आई एम मेंशनिंग दिस टर्म cell cycle. What does it mean? It does not mean only mitosis. It also means includes replication. Now, based on biochemical events, four phases are there. Biochemical, not cytological events. So four phases are there. ये दोनों हमने अभी बात की. Replication in the S phase, mitosis in the M phase. These two are active phases of cell cycle. And then there are certain gap phases also. G1 before S phase, we won't talk about this at the moment. G1 before S phase or G2 above before M phase. So these are intervening gap phases. We are going to talk about this now today. This is with reference to p53, more important. This is called G1 to S में जब transition होता है. This is actually what we call a checkpoint, a control point. So what do we mean by control? What do we mean by checkpoint? You see, the cell makes all or none decision late in G1 here. As the figure shows, late in G1, the cell makes all or none decision whether to go to S phase or remain in the G1 phase. और तब तक ये G1 phase में रहता है till every damage has been repaired. और अगर एपोप्टोसिस की जरूरत है तो वो भी हो जाता है. तो this all or none decision is made. So this all or none decision, this defines a checkpoint. So all or none decision, S phase is embarked upon only when the cell is ready, after any damage is sustained earlier has been thoroughly repaired. So this all or none decision, this defines a G1 checkpoint. So it is actually, you can also call it damage checkpoint. And we are going to see in the next slide if what decides कि G1 to S transition होगा या नहीं होगा. Cyclin T. Cyclins actually they are a group, these are a group of proteins which make periodic entry during the cell cycle. Appear, disappear. G1 checkpoint पर जो cyclin appear होता है, that is called cyclin D. So cyclin D, this determines कि G1 से S पर जाएगा या नहीं जाएगा. Rather, it promotes entry, this transition, crossing of this checkpoint. And cyclins, they talk along with certain enzymes called cyclin-dependent kinases. All right, cyclin-dependent kinases. And this is a complex series of events I'm going to talk in end of this lecture. अभी आपके लिए इतना ही काफी है कि cyclins are promoting G1 to S transition, crossing of G1 checkpoint. What about p53? p53 inhibits action of cyclin D. तो इससे क्या होगा, साइक्लिन डी का इनहिबिशन हो गया, दिस प्रमोशन will be blocked. As a result, G1S checkpoint will be blocked, right? So that is how cell cycle arrest is caused by p53 acting via cyclin CDK complex. तो ये तो हो गया first point, cell cycle arrest. What about the next two, repair and apoptosis? ये कैसे होता है? Repair and apoptosis. As we said earlier, p53 drives expression of a number of enzymes which are involved in DNA repair, which are involved in apoptosis. We also said that the half-life is 5 to 6 minutes and intranuclear level is very low because it is very unstable molecule. ये तो जैसे ही प्रोड्यूस हुआ 5-6 मिनट्स में ये ब्रेक हो जाता है. So its level must be increased following DNA damage. How does that happen? We are going to see now. This DNA has been damaged as shown by these arrows. Now this causes stimulation of a kinase called sensor kinase. Sensor kinase को stimulate हो जाएगा. तो these sensor kinases, जैसे कि नाम से पता लग रहा है, ये DNA damage को at the site of damage they will sense it and they will relay the signal further, in the process activating some other kinases called checkpoint kinases, abbreviated here checkpoint kinases. तो the signal DNA damage का relay हो चुका है. These checkpoint kinases will come into action. They cause, okay, let me before said, p53 की half life short, उसका क्या reason है? उसका reason है there is a proto-oncogene called MDM2. It very rapidly binds with the p53 and sends it for degradation to proteasome. तो इसलिए p53 की हाफ लाइफ बहुत कम होती है. So p53 is very unstable because MDM2 इसको टिकने ही नहीं देता, इसको फटाफट बाइंड करता है and the complex goes to proteasomes for degradation. तो चेक पॉइंट काइनेज क्या करता है? Checkpoint kinase you people know very well, intermolecular shift. तो kinases ATP से phosphate group shift करते हैं. They transfer the shift phosphate group to p53. तो ये on as many as 12 sites p53 gets phosphorylated. Now an important point, phosphorylated p53 is a poor substrate for MDM2. Simply stated, MDM2 which rapidly bound with a p53 after it has been phosphorylated, MDM2 will not be able to bind it that well. Alright, so p53 will not be promptly sent to the degradation, तो इसकी half life इसलिए increase हो जाएगी. तो हमने देख लिया p53 की हाफ लाइफ इंक्रीज हो गई, सेल साइकिल अरेस्ट भी हो गया था एंड नाउ एडिक्वेट टाइम फॉर रिपेयर इज अवेलेबल. अब लास्ट में हम देखने वाले हैं मैकेनिज्म ऑफ p53 एक्शन. ये मैंने बोला था कि p53 कैसे करता था. p53 was inhibitor of cyclin CDK complex, और cyclin CDK complex हमने जैसे बोला था, this is normally a stimulator of the G1 to S transition. तो ये कैसे cyclin stimulation करता है, let us see. This is the point we are going to see now. I hope I am able to make myself clear कि प्रॉब्लम क्या है. प्रॉब्लम ये है, हमको मैकेनिज्म देखना है कि कैसे ये सब हो रहा है एट द मॉलिक्यूलर लेवल. You see, there is a transcription factor. Let me outline the mechanism in further details called E2F. It's a transcription factor. And every transcription factor has got a domain which binds the represented as for my fingers, for example, which binds the DNA and enhances transcription. और ये E2F, it is such a potent transcription factor. It enhances synthesis of more than 500 proteins, among them some enzyme, DNA polymerase, tetrahydrofolate reductase, and several cyclins. और ये सब मिलकर चेक पॉइंट को क्रॉस करने में हेल्प करते हैं. But normally, in a quiescent cell, in a silent cell, E2F is under inhibitory protein, under influence of an inhibitory protein called RB protein, RB retinoblastoma protein. ये E2F को बाइंड करता है और देखो ये फिंगर्स डीएनए बाइंडिंग डोमेन मैंने बोला था, उसको मास्क कर देता है, उसको हाइड कर देता है. तो व्हाट विल हैपन, E2F का डीएनए बाइंडिंग डोमेन हाइड हो गया. एज अ रिजल्ट, in a silent cell, E2F will not be able to encode the protein और G1S चेक पॉइंट क्रॉस नहीं होगा और G1 फेज प्रोलॉन्ग होती जाएगी. ऑलराइट, अब साइक्लिन क्या करते हैं? साइक्लिन D and CDK, एज आई सेड, फॉस्फोरिलेशन करते हैं. काइनेजेस किसका करते हैं यहाँ पर? E2F का और RB दोनों को फॉस्फेट ग्रुप अटैच कर देते हैं, नॉट एट अ सिंगल साइट बट एट मल्टीपल साइट. और बिल्कुल सिमिलर मैकेनिज्म से जब फॉस्फोरिलेशन हो गया तो इनकी बाइंडिंग वीक हो जाएगी. तो देयर इज फॉस्फोरिलेशन एंड dissociation. In other words, cyclins are removing the inhibitory effect of the retinoblastoma protein. I am repeating, retinoblastoma जिसने E2F को पकड़कर जकड़कर इनएक्टिव करके रखा हुआ था, now E2F is set free because of phosphorylation. तो ये आजाद हो गया पंछी, यहाँ पहुंच गया, and after that G1S progression will occur. So p53, let me complete it. ये फिगर कंप्लीट कर दें. p53 कैसे कर रहा है? हमने देखा साइक्लिन प्रमोट कर रहा है, इसका इनहिबिटर p53, तो सेल साइकिल अरेस्ट कर देता है. तो हमने ये डिटेल मैकेनिज्म में इसका देख लिया. तो फॉर इंक्विजिटिव माइंड्स, इफ यू फाइंड इट इंटरेस्टिंग, you may retain it. All right. So summary of events. So summary of events, ये repetition है. आप अपना flow diagram खुद बनाइए. Don't rely on this. p53 inhibits cyclin CDK complex. All right. ये क्या करता है? Phosphorylation and CDK. ये phosphorylation is prevented. फिर क्या होगा? E2F remains under inhibitory control of RB and everything is stuck at G1 checkpoint. All right. So finally, you should be able to answer this question. Why p53 is guardian of the genome? Because it is key integrator of DNA damage response. Another question for you. RB, retinoblastoma protein, that is called guardian of G1 checkpoint. तो उसका आंसर आप अपने आप देखिए उसको, बिकॉज G1 checkpoint पर वो ETF को पकड़ कर रखता है, एटसेट्रा. सो आई वांट कंप्लीट So just a trifle in a world full of carcinogens, be someone's p53. Another, just uh, I'm reminded of an adage inspired by some, American society. I'm told he, one should have courage to change the things which he can. One should have patience to accept the things which he cannot, and wisdom to discriminate between the two. So our cells, they have got this inherent wisdom and patience and courage also. They change by repair. They accept which they cannot repair. All right. And discriminate between the two. सेल जिसकी रिपेयर हो सकती है डीएनए की उसको कर दिया, जिसको नहीं कर सकते नहीं. I find this particularly interesting, isn't it? Now, just one or two points. Why p53 and RB? They are called anti-oncogenes, encoded by this and this chromosomes. Why MDM2 should be called proto-oncogene? And one additional point, anti-oncogenes are always more potent compared to proto-oncogene. जैसे आपको पता है क्रोमोसोम्स पर होमोलोगस दो कॉपीज होती हैं. इफ देयर इज म्यूटेशन इन वन, सिंगल हेट्रोजाइगस म्यूटेशन, वो एंटी ऑन्कोजीन में इफेक्ट नहीं डालता. ओके, रीड अबाउट मोर अबाउट इट. We are going to discuss in the next lecture. This is Lie-from-Lie syndrome. This is when a single copy of p53 is mutated. All right. So such people, they are much more prone to develop cancer in their future life compared to the normal person. So thank you very much, and